Your lab at MIT was the first to identify sirtuin as the gene that controls aging in yeast cells. And your lab was also the first to discover a link between NAD and aging by showing that sirtuin and related proteins function as NAD dependent enzymes. And that means that they need NAD to function. But one of the most compelling components of this discovery, I think, and the audience really agreed, is how you go from controlling aging in yeast and translate that to a fundamental process of aging in humans. So even though you really explained this incredibly well in the webinar, people still had some questions about how do you make that leap. So maybe you can start by explaining kind of some basic aspects of how research works, and then how in this specific instance you went from aging in yeast to aging in humans.
Yes, research generally is best when it really asks a specific question with a specific focus. And the way we started this was to ask the question, is there any gene in yeast that is particularly important in controlling aging? And so the focus was on yeast aging. And what emerged from that study was a gene called SIR2, which was a positive regulator of lifespan. The more activity SIR2 in a cell, the longer the lifespan in yeast. So the way to extend that result to other organisms or to test, is this gene important in aging in other organisms, is you have to hope, and this turned out to be true, that other organisms have an equivalent gene. Okay, and SIR2 is a highly conserved gene. So we could ask the question, would the SIR2 homologue in another organism, for example, the roundworm, function in the same way? And that's what we did. We simply uh, turned the lab over to studying worms for this project, and had a clone of the C. elegans SIR2 gene. And we also then made extra copies of SIR2 in worms and asked if the worms lived longer, and they did. And that really told us that this function was conserved. Now, we can't say for sure that, SIR2 is doing it for the same reason in the worms as in the yeast. But what we do know is it has this intrinsic biochemical activity of an enzyme, and it's an enzyme that requires NAD. So we infer that there is a relationship between the mechanism in yeast and the mechanism in the higher organism, the worm. Now, in yeast, what we really think is important is the enzyme acting on histones, the proteins that are bound to DNA. And this could well be the universal truth in all organisms. And what it does in yeast is it changes a modification on these histones in a acetyl group by removing it, and that promotes a change in the structure of the DNA protein complex so that more genes get silenced. So, what it does when it's most active is the SIR2 gene promotes an epigenetic change in the genome in the yeast nucleus. And we speculate that that is at least one highly conserved mechanism that would apply to higher cells, higher organisms as well. So once we knew this was conserved, its effect on aging in yeast and worms, there's a very strong inference that it's conserved elsewhere as well. And so we started to look, and this became, of course, a 10-year odyssey, because we wanted to ask the question in mammals. And the mammalian system that is most valuable in the lab is mice. And so we did the same kind of experiment in mice and confirmed that in mice as well, SIR2 was involved in mediating effects of diet on aging, this calorie restriction, which is known to be an important component. And another lab, Haim Cohen, showed that one of the SIR2 homologs in mice, and mice has 7 homologs, as do we, to the yeast SIR2 gene. And a particular one of those 7, called SIRT6, could make mice as animals live longer when this gene was super activated in a transgenic mouse, a mouse that had an extra copy of the gene. So that really ties it up and says that SIR2 and its homologs, which came to be called sirtuins, these proteins are involved in longevity across a wide swath of nature, likely in every organism.
The leap from yeast to humans didn't happen all at once. It wasn't even in that 10-year odyssey. It took quite a bit more time, and that link really starts with NAD. So, can you talk to us a bit about that, Lenny?
Um, we were interested right about the same time we were doing the yeast work, a little bit after, what the actual activity of the SIR2 protein was. And at that point, as I said, we knew that SIR2 genes were conserved. That is to say, other species had very similar genes to SIR2, which encoded similar proteins with a similar sequence. And so that enabled us to purify the yeast SIR2 protein and the human SIR2 protein. So we had these two reagents in hand, and this is in the 1990s still. And we set about to discover what they did, and that's when we found that they had this unique enzymatic activity. They were proteins that deacetylated other proteins, so that's the enzymatic activity to remove acetyl groups. But the key thing was that it required a compound called NAD, which is present in all cells and is involved in energy production in the mitochondria. So it's a very important small molecule in cells, has a molecular weight of about 1,000 daltons. And it's known to be involved in a few processes in cells, the most important of which is to generate energy in mitochondria. So the fact that these SIR2, these anti-aging proteins, required NAD was very, very interesting. Okay, and that was always at the forefront of our thinking as we were doing these experiments in mice. Okay, so what it turns out, NAD is really the conduit that brought us from yeast to humans. And the way that worked was it was shown first in mice by Shin Imai that NAD levels actually declined in cells as mice got old. So old mice had lower NAD than young mice. This is very provocative because that means that the activity of the sirtuin proteins, which require NAD, would go down with aging. And it also meant that you might be able to restore it by adding NAD or a related compound to an old organism. Okay? So that really set the next stage of discovery. And it was really with Elysium that we were able to kind of fulfill this path because it turned out that the NAD precursor NR, nicotinamide riboside, is a very good molecule to use. First of all, you can get this compound very pure, and it gets into cells, and the cells will convert it into NAD. And it works much better than, say, NAD itself, which really doesn't get into cells, so it doesn't work. So we were able at Elysium to conceive of a product, our first product called Basis, which includes NR in a pill. And at Elysium, we've done a lot of studies in the ensuing years, about 10 years now, and other research centers around the world have done human trials, and there's a tremendous amount in progress right now studying the effects of boosting NAD in aging people. And what I can tell you in summary is we know that they're healthier. Okay, I don't think I don't want to say with any certainty that we're sure they live longer. although we think that aging might be slowed down by using these biological clocks that exist. This is a bit of a tangent, but there's some evidence, emerging evidence, that aging is slowed down. But we know that it confers health benefits. And I'll just name one. There was a recent study done in Copenhagen on COPD in collaboration with us, Elysium, that showed that the inflammation associated with COPD due to pro-inflammatory cytokine was reduced by treatment in these people. So we think that this has the potential for having broad therapeutic effects in people, but also slowing down aging. Now, what really ties up the whole package in a nice ribbon is recent studies on a human gene called SIRT6. So I mentioned earlier that there are 7 SIRT2 homologs in humans. One of them is SIRT6. And what was done is uh, groups who study, human geneticists who study aging populations of people, looked at many genes to see if there were any what are called polymorphisms in the genes, which are stably inherited forms of the gene, which slightly differ from the most common forms. And what they looked for was polymorphisms in all the genes that are known to be related to DNA function and repair in cells. It was about 500, I think. And one of those 500 in the bin was SIRT6. And what they found was that there's a very interesting, uh, what's called an allele or polymorphism, if you will. These are human genetics terms in SIRT6 that is fairly rare in the population, but is considerably enriched in people who live to be 100. These people called centenarians. So that's a very interesting finding, suggesting that having this allele of SIRT6 is associated with extreme longevity in people. And this is just really just beginning. We just know the tip of the iceberg of this story now, and we'll know more in a few more years. But it really does seem to be the same idea, harking all the way back to yeast, where if you increase the activity of a sirtuin in an organism, you can extend the lifespan, or promote longevity in the case of these people. So, that's how you kind of bridge this gap, and it's a big gap between yeast and humans. And of course, you can't assume that something that's true in yeast or even mice will be true in humans. You have to do the studies, which is why I've been a strong proponent of testing in Elysium in humans to do real human trials to establish things you think might be true, to establish them scientifically.